Okay. All right. Thank you very much. Hello, everyone. So thank you, Kara, for the introduction. So this lecture today, we're going to be discussing genetics in epilepsy, and I had the supervision of Dr. Danielle Andrade. For those who don't know her, she's the director of the Epilepsy Genetics Program, and she helped me with this presentation. So I'm going to start going back to basics. So first, I'm going to try to define what a few things are, like the DNA, what genes are, what mutations are, before we can understand how that plays a role in epilepsy. So first of all, what is the human DNA? So DNA actually stands for deoxyribonucleic acid, and it's made of units of biological blocks that we call nucleotides. So if you look at this image, you see the colorful blocks that we have: adenine, thymine, cytosine, and guanine. So those are the blocks of the biological blocks that I spoke about, and they are what form the human DNA, which you'll also see in a representation on that image. And the DNA is what contains our hereditary material and also our genes. And so therefore it's what makes us so unique. So the DNA, the complete set of our DNA is what we call a genome. So that means our whole DNA. And it contains a lot of genes. So it has 20,000 genes and also 23 pairs of each cell will have 23 pairs of chromosomes. So 23 will come from the mom, and 23 will come from the dad when a baby cell is formed. So we see on that image that 23 chromosomes are coming from the egg and 23 are coming from the sperm, and they're forming the baby cell with 23 pairs of chromosomes. So as I said, half is going to be inherited from each parent, and the DNA is going to be packaged into those pieces that we call chromosomes. So I haven't explained before, but what I showed before on the DNA, it's actually tangled up together and it forms the chromosomes. So instead of having a very long piece of DNA, it's going to be broken into those 23 pairs. And how about genes? What are genes? We're going to talk about genes a lot in this presentation. So the genes, they are a sequence of the DNA. So they are a portion of the DNA that's going to determine different proteins in our body. And those proteins, they're going to have a variety of functions and will determine how we develop, how we grow, how we function. So each organ will have their specific proteins. Everything is formed by proteins. So as we see on this figure.
Sorry, Dr. Marcus, I think I've lost your sound. I'm not hearing you.
Sorry?
I'm not here. Oh, yeah. I'm now hearing you. Went blank for a bit. I wasn't hearing you.
Okay, sorry. So we see the genes, and then the genes are going to be read by what we call RNA, and then they're going to be transformed into those proteins that are going to be part of everything we have in our body. And with a few exceptions, everyone will have 2 copies of every gene. So one gene will come from each parent. And those genes, they are actually similar from person to person. So when we map the human DNA and the genome, we actually see they're very similar. But there are some variations. And most of the times, those variations, they're not going to cause any impact on that person. They're not going to cause any disease. But sometimes when we have some variations on these genes, it's what we call a mutated DNA or mutated gene. Then we can have some medical issues such as epilepsy. Okay, so this is just a summary of what we talked about. So we have in our body many cells, Each cell will have 23 pairs of chromosomes, which is the tangled DNA. And each chromosome will have many genes. And if we have an abnormal gene, then that's what we call a variant or mutation that we're going to be talking a lot to here. So I think one thing that is very important to define here is epilepsy genetic? So this is the whole purpose of this presentation. So this first figure is something we have, this is the knowledge we had in 1985. And what we knew back then is that approximately a quarter of the epilepsies, they had some identified cause for them, such as the ones you see there, like trauma, stroke, tumor, infections, and among others. And the rest, the 75% of the rest, they were what we call idiopathic. What does that mean? Idiopathic actually means we don't know the cause yet. So what happened over the years is like, next, please. What happened over the years is that this second figure is what we call the 2014 paradigm. So this is what we know now. What we know is that approximately a quarter of the epilepsies have other causes, like the ones that I showed before, like trauma, stroke, tumor, and the rest, almost 70% of the rest are what we consider genetic epilepsies. So nowadays we know that a lot of the epilepsies are actually caused by genetic issues. And since we define that, one confusion we have with that term genetic is that if that means the same as inherited, does it actually mean the same? So actually, no, those two things are different. So when we say an epilepsy run in a family, that means that epilepsy is inherited, but also means that's genetic. However, can you put the next one, please? So this next figure shows a type of epilepsy that is inherited. So you see the people in blue, they are the ones affected by the epilepsy. And you see that runs in the family. So that's something we consider, that is an epilepsy we consider inherited and also genetic. Next one, please. Now this next figure, this next image, actually shows that there's only one person affected and the parents are not affected. So in that case, we say that this mutation, this genetic abnormality, is not inherited. So what happened in that case? What happened is that those cells, that individual DNA, mutated, became abnormal when this individual was developing in the uterus of his or her mom. So that's what we call it de novo mutation, which means it arose without coming from the parents. It happened after conception. Next one. So if epilepsy can be inherited in some cases, what are the chances of actually inheriting epilepsy? So first-degree relatives of a person with epilepsy, and by first degree I mean parents or children or siblings, they are going to have a 4.7% chance of developing epilepsy prior to the age of 40, which is 3.3 times higher than the general population. And that actually depends on the type of epilepsy we're talking about. So for example, if a person has what we call a focal epilepsy, which is when a single area of the brain is causing the seizures, like the representation on the image, then that risk of having another relative involved with the same disease is 2.6 times higher than the general population. However, if this epilepsy is what we call generalized, which is the other figure on the bottom, which means the whole brain is causing the seizures at the same time, then this risk is going to be higher. So it can be 8.3 times higher than the general population. So it all going to depend on the type of epilepsy and what the type of gene that we're talking about. This one. So another factor that also plays a role in determining the risk of a person inheriting epilepsy is whether the gene that we're talking about is what we call autosomal recessive or autosomal dominant. So I'm going to explain what that is. So an autosomal recessive disease means that disease only appears when the individual has received 2 copies of the abnormal gene. So like we see on this image, you see in purple what we call a variant, which is when the gene is abnormal. So each parent has 1 copy of the variant, and they're gonna pass it along to their kids. So the kid who got both the purple variants actually is affected by epilepsy. So that's what we call an autosomal recessive, which means that each parent has to give one gene to the child in order to have the disease, and the parents are not going to be affected. Okay, and if you are a person that has autosomal recessive epilepsy, what are the chances of having a child with autosomal with the epilepsy? So in that case, you would have to have a child with someone who also has another gene mutated, because since you're only passing along one abnormal gene, you would need to be married to someone that would also have an abnormal gene in order to have children with the same disease. So it's more difficult to transmit that to your offspring if you have an autosomal recessive disease. Okay, and this is usually what happens in consanguineous marriage, for example, when we have relatives getting married and they have a similar DNA, and therefore we have 2 people that get married and they have the same genetic abnormality. Now, the other type that we have is what we call autosomal dominant. And in that type, we only need 1 abnormal gene to cause the disease. So we see there the dad it could be either the dad or the mom, but the male is the one that has the variant, the abnormal gene. So for each kid that he passes the abnormal gene, that child is also going to have the disease. So the chances of transmitting that disease are much higher than the autosomal recessive, which was the previous one. So in that case, the chances are actually of 50% of having children with the disease, because you only need 1 copy, right? Next. Okay, so moving on, are there epilepsy-related genes, now that we're talking so much about genes? So actually, the first few epilepsy genes, they were discovered in the late 90s, so it's quite new. And there's been a lot of advances in DNA sequencing that have now identified hundreds of genes that are related to epilepsy. So it's a very new and growing field. And new genes are being identified on a regular basis. So it's always been updated. Some of these genes, they are identified in patients that only have epilepsy, and they are very specific to epilepsy. And some of the other genes, they are also related to other diseases, like other conditions, like developmental delay, autism, and intellectual disability. Next one, please. Next one, please. Okay, thank you. I think the previous one, please, Carol. I think you passed more than one. Okay. So in terms of genetic tests, what type of tests do we have? And there are many types of genetic tests, and that's why it's so important for us to understand a little bit of how the DNA and the genes work, because as I explained before, we can have tests that will look at individual genes. So for example, I want to test a specific gene for epilepsy. You can order that specific one, or you can test groups of genes. So you can order what we call an epilepsy panel, for example, that will have many different types of genes that are related to epilepsy. Or you can look at the chromosomes. So you can look with a larger, like as if you were looking from more above, like you're looking at the big picture instead of the small genes. And because there is no single test that can diagnose all genetic epilepsies, multiple tests might be recommended. So it's not uncommon for us to see one test being ordered after another, because as I said, there's not one specific one that's going to look at everything at the same time. And there's also variation between the labs that will do these tests. And there's considerable, as I said, considerable variation between them. So why all of this is so important? Because we have so many different types of tests, and those tests need interpretation, it's important to have a healthcare provider that is knowledgeable about genetic testing, involved in the process of ordering and interpreting those results. Another question that patients will usually have is, how is genetic tests performed? It's actually quite simple. Most of the times, we just have a blood sample. In some cases, we can have a saliva sample or a cheek swab. And less frequently, we might order a skin biopsy, but that's really a rare case. And once I get a genetic result, what type of results can I actually get? So you can get a positive result, and that means that genetic change that was found is actually the cause of your epilepsy. And a positive result in a healthy individual otherwise can indicate that that person has a risk of developing epilepsy, even though doesn't have epilepsy yet. So that's what it would mean in a healthy person. You can also get a negative test result, and that means a genetic cause was not found for that individual, but that does not completely rule out the possibility of a genetic diagnosis. Because as I said, we're still discovering a lot of genes. So the fact that you have a negative result does not mean your epilepsy is not genetic. Maybe it means more testing might be considered and that has to be reassessed in the future, but does not necessarily mean you don't have it. This one. You can also get a result that's called uncertain, which is quite frustrating because an uncertain diagnosis actually means something was found. So a genetic finding was detected, but the meaning of that is not yet understood. So what does that mean? It means they found a mutated part of the gene, like a variant, but they don't know how that plays a role in that individual's disease. So what happens a lot of the times, we have to look again at that test result a few years later and then see if that was reclassified. Because as I said, since it's a growing field, what happens a lot of the times is that those genes, they get reclassified and reinterpreted later as, for example, being benign, which means that test result is negative, or pathogenic, which means that test result is actually positive and that gene is responsible for the disease. So that's what an uncertain test result means. It means we have to further reassess that in the future. Oh, previous one, please. I think there was one before. Okay. So should every person with epilepsy get a genetic test? So As I showed on that on the previous one of the previous images, up to 70% of the cases can be genetic in people with epilepsy. But if a person already has a known cause for their epilepsy, such as a brain injury or a stroke, genetic testing might not be indicated because it's not going to tell us much. It's probably not going to find anything. However, if a person doesn't have a known cause yet, it is indicated to get genetic testing. And it's more likely that you're going to find something positive if that person has a type of epilepsy that is very difficult to treat, which is when we call it refractory, or if that individual also has other neurological or neurodevelopmental issues, such as developmental delay, autism, or a movement disorder. And also in cases where there's a strong family history, then there are higher chances of finding a genetic cause. And if I do get tested, does it mean my family members need to get tested as well? So a positive result on a genetic test might as well impact family members. So sometimes it is important to test the family members. When we do initial tests, usually we might extend to first-degree relatives, such as parents or siblings. But depending on the results, more distant family members might also benefit from that testing. And whether it's recommended or not to test family members, it will depend on the type of genetic disease we're talking about. It will depend if it's inherited or not, because as I said, sometimes it's not inherited, it's de novo. In such cases, it's unlikely that other family members will have the disease. And it's also important to know what type of inheritance we're talking about, as I explained, if it's autosomal dominant or if it's autosomal recessive. So it is important that individuals and families seek guidance from a medical geneticist or a genetic counselor to find out if testing is important or not. Next one, please. And lastly, I would like to discuss a little bit why is genetic testing so important. So genetic testing might help identify a cause of a person's epilepsy. It may lead to an accurate diagnosis and therefore a better medical management. And it might be particularly helpful in patients that are not well controlled of their seizures. And that can really change the way we manage the medication and how we're going to continue with the treatment. That can also give us more information about long-term outcomes. So for example, if we have an adult that has a type of epilepsy that started in childhood, that we know that gets better with age, and that person is seizure-free, we might consider stopping the medication because we have that data from previous patients that also have that same genetic mutation. So it's very important for us to understand what type of disease the patients have in order to help predicting what are going to be the outcomes in the future. That might also help to better inform family members of their own risk and also their chances of having a child with epilepsy. So for example, if you have a patient with epilepsy and its sibling wants to have children, maybe that sibling would like to know if they have a chance of having a child with the same disease. It also makes it possible for individuals and families to access other resources. So for example, advocacy groups and peer families, which are very important. It may also lead to participating in research studies that are specific to that genetic diagnosis. And finally, it might help putting an end to what we call a diagnostic odyssey, which is a search for an answer. So a lot of the times what patients need is just an answer of what is going on with them, what is the cause of it, and what it's going to be like in the future. And lastly, what I would like to talk about a little bit is about precision medicine and gene therapy, because this is really the future of genetics. So this is what we are uh, going through now and what we aim in the future. So precision medicine basically will combine everything we know about a specific disease and the genetic code and try to aim the treatment to that genetic code. So it helps us to tailor the treatment and makes it more targeted to that genetic disease. And we will eliminate a lot of the guesswork. So sometimes we have to start a treatment, but we don't know if that medication is the best one for the patient or not. And by knowing what genetic abnormality they have, we're going to be able to choose better what type of medication we can give to the patients. So a few examples that we have, I don't know if anyone has heard about it, but there is a syndrome called Dravet syndrome, which has patients with very difficult to treat epilepsy. And in such cases, there are two medications that are specially recommended for them. One is stiripentol, and the other one is called fenfluramine. And it's also recommended to avoid certain medications. So this is the type of information we can get from getting a genetic result. Another example is a disease we call GLUT1 deficiency, where the ketogenic diet is actually the best treatment for those patients. And finally, gene therapy. So gene therapy is also considered precision medicine, and it's when we are actually able to modify the genetic code, the genetic makeup of a cell, in the way that will treat the disease. So everything I showed before, when we have the genetic variant, the gene abnormality, it's like something like the medication, the treatment that they're going to target, use, is going to go there on the DNA, make some changes there, and then that gene will not be defective anymore, and the protein will start to be produced in a regular way. There are many ways of doing it. It's a bit complex, so I'm not gonna explore that further. But basically, by changing the genetic code, we're able to modify the disease progression. So it's a pretty revolutionary way of treatment, and it's been developed for many diseases, not only in epilepsy, but many other areas in medicine. This is an example, for example, of an example of a drug that was approved for an epilepsy that's caused by Batten disease. It's a rare form of epilepsy, and they have a drug that was approved as an orphan drug that actually modifies the genetic material. So it's pretty important to know that such things are being studied now, and there's a lot of research going on in that field. So in summary, what we have is that genetics in epilepsy is a new and emerging field. For some individuals, it's not yet possible to determine whether their seizures are due to genetic factors, but that has been changing over time, and more genes are being discovered every day. In many cases, the researchers are just beginning to learn about specific genes and the types of epilepsies associated with them. So it may be hard to predict an individual epilepsy long-term outcome because we don't have a lot of studies, so more research is necessary. But the good news is that a lot has been done in that field. And the good news too is that genetic testing is now widely available for both children and adults. So OHIP, for example, covers for most of the genetic tests we have, as long as it's well ordered and there's some genetic counseling. So a genetic diagnosis might provide valuable information about the long-term outcome and the treatment for those patients like I showed. There's a lot of ongoing research efforts to learn about genetic changes that may lead to epilepsy. And this knowledge will help us bring new and better treatments for individuals that live with epilepsy. And finally, I just want to leave some resources here. So A lot of this presentation came from material from Epilepsy Foundation. It's an amazing resource. It has pretty much everything there explaining about epilepsy genes, among other epilepsy-related treatments. I also like to point out that this website, for the third one for clinical trials, has all the clinical trials for patients with epilepsy, and some of them would involve gene therapy, for example. And also the Rare Epilepsy Network, where groups of families and patients with rare diseases such as some genetic epilepsies, they might be able to connect with each other and learn more about their diseases. Thank you very much.